Hello student, today we are going to study on types of antigen antibody reaction. In previous lecture we are discuss about what is the antigen antibody reaction and what are the requirement of antigen antibody reaction. Now what are the types of antigen antibody reaction? That is precipitation reaction, agglutination reaction, complement fixation, ELISA and immunofluorescence. One by one we are taking the brief discuss on it. Now first of all, that is a precipitation reaction. The name suggests that there is a precipitation, means what? There is a some kind of the PPT or precipitate is formed. When there are the two image, that is one is the antigen and another one is the antibody. When a soluble antigen combine with its antibody, specific antibody with the presence of with the presence of an electrolyte, that is sodium chloride, and this reaction is carried out at a particular temperature and pH, then it forms an insoluble precipitate that is called as the antigen antibody complex, and this antibody causing the precipitation is called as the precipitin. There is the particular precipitin or thick PPT is formed, and that is called as the precipitin, and the reaction is called as the precipitation. Let's see in the diagram. There is the antigen antibody complex is formed. After that, that red color antigen will be destroyed by that specific antibody. Now the next reaction is the agglutination reaction. Agglutination means it is called as the clumping of particular antigen. That soluble antigen will be destroyed by antibody, and there is the clump or clump the particle or agglutinate the particle, and that particular reaction is called as the agglutination reaction. So when a particular antigen is mixed with an antibody in presence of electrolyte with the suitable temperature and pH, the condition is same over here also. So the particle are get clumped and agglutinated. So the antibody of the serum causes the cellular antigen to form clumps. The cell of antigen will be clump, and that is called as the agglutinins. And the particulate antigen are aggregated and are termed as the agglutinogens. Okay. So there is the one method that is called as the slide agglutination. So it is the rapid method to determine the presence of agglutinin antibody. Let's see. So in a particular reaction, there is a soluble antigen with antibody will be placed in that particular drop and then mix with the loop, fine loop, and then it is then there is the destruction of that particular antigen will be done, and clumping of antigen is occur, and that particular solution will be take on the test culture, and that particular colony of microorganism or viruses or bacteria will be seen. Now next, so to a uniform suspension of particulate antigen, a drop of saline is added to drop of antiserum. Means what? It is for the mixing of that antigen and antibody properly. So the slide is gently rock and fine loop is used to mix the content, and if granulation or agglutination is occur, means the test is positive. So it take a minute for the complete the reaction visible to the naked eye, and sometime confirmation may be done by observing the slide under microscope. It is useful for generally for blood grouping, hemagglutination and cross matching also. Then this is the hemagglutination inhibition. So first is the patient in first diagram, that is a patient sample, that is human IgG expression, that is G1 epitope, and with the RBC. Then there is the no agglutination, means that test is the A positive reaction. Then another one in case, that is human IgG with the heavy chain isotope and patient sample means, and then IgG. So there is the agglutination. Agglutination occur means it is the A negative reaction. So next is the first is the slide agglutination, then next test is the tube agglutination. Now what is the tube agglutination? So it is the standard method for the quantitative estimation of antibody. Means what? In a particular sample or in a particular serum, what is the amount of antigen and antibody is present, and on the basis of that we will conclude about the particular criteria of that disease, or a particular antigen is present in that solution. So the serum containing the antibody is diluted serially with the saline, and in several small test tube. So with a constant volume of antigen suspension is added. Then the control tube is also kept over here with the no antiserum, and these are incubated with the visible agglutination is observed in that reaction. So tube showing the highest agglutination is referred at the titer. Titer value means what? It is the highest agglutination constant. So tube agglutination is employed for the serological diagnosis, that is typhoid, brucellosis and typhus fever. So Vidal test is used for the estimation of typhoid fever caused by the Salmonella typhi in that particular, this is the Vidal test. So 1:20, 1:40, 1:80. Then titer value is the 1:60, means at that condition there is the, if 1:60 is the titer value is observed, means that particular patient or that particular person is suffering from the Salmonella typhi infection. Beyond that, that is 1:360, 640 and control is normal, negative. So that 320 and 640 it is showing about the extension, or it's showing the particular concentration of that particular antigen present in that or individual. But 1:20, 1:40 and 1:80, this is the normal condition, and it will be concluded on the basis of the titer value. Titer value is taken as the constant or it is taken as a reference. So it is the Vidal test. Now next, that is the complement fixation test. So lysis of RBC or bacteria require the some non-specific unstable component or fresh serum which are called as the complement. So complement are the special protein which are responsible for the antigen antibody reaction, and complement system comprise about the 11 protein and that are present in either every individual. So that bind to the Fc component, fragment of crystallization component of antibody, and which involved in the antigen antibody complex. And these ability of antigen antibody complex to fix the complement, it is used for the complement fixation test. What happened here? In the first stage, the antigen with antiserum is heated to first 56 degrees centigrade to inactive the complement, and then that mix in presence of the amount of complement, and that is known amount of complement is mixed with that particular antigen antibody complex, and it is incubated for the 4 degrees centigrade for the 18 hours. Then after that, the antibody specific for that antigen is present in the serum, and then antigen antibody complex will form that will fix the complement. Let's see in the first diagram, that is first diagram. In that there is a, this is serum with that antibody, then that antigen is added to the bind to with that antibody, then known complement molecule is bind with that antigen antibody complex, then there is the hemolysis of sensitized RBC with the serve as the indicator that RBC is settle into the palate, means there is the no lysis, and this is the reactive. Let's see, there is another test. There is serum with the antibody, then unbound antigen. Unbound antigen is there, means there is the that particular antibody will not form the antigen antibody complex. And then what? There is the unbound. Then there is the unbound complement is there, and hemolysis of sensitized RBC serve as the indicator, and RBC lies by unbound complement, that is lysis. So that is the non-reactive reaction, means it is the false reaction, and it is the reactive reaction, and this is complement fixation test. Now next test is the enzyme link immunosorbent assay, that is ELISA. It is used for the detection of the AIDS also. So ELISA in 1971, that is HIV इन्फेक्शन के लिए इसको यूज़ किया जाता है, HIV इन्फेक्शन टेस्टिंग के लिए. So in 1971, enzyme labeled antigen and antibody were developed as the serological reagent for the assay of antigen antibody reaction. So there are the very simple and sensitive and economic hazards when compared to the RIA. So ligand used here with the molecule which can be detect the antibody. It is conveniently coupled with an enzyme such as peroxidase enzyme, beta-galactosidase enzyme, alkaline phosphatase enzyme. Then So there are the three types of ELISA, that is indirect ELISA, sandwich ELISA and competitive ELISA. Now what is the indirect ELISA? So this technique is used for the detection of HIV, and these envelope protein are developed by the recombinant DNA technology and coated on the surface of microtiter plate. So suspended serum is added and unbound protein were washed off. Then second is the sandwich ELISA. It is also detect in the presence of the antigen in a sample, and the well is coated with the antibody specific to the antigen, and then suspect serum is allowed to react and the well are washed to remove the unbound antigen. Take a brief account with the help of diagram. Now first is the indirect ELISA. So there are the antigen coated in a well, then it is washed, then specific antibody are adding to the reaction, then it is washed with the saline sample. Then other enzyme will be conjugated, means secondary antibody is again add here. Then wash means what? At that color is yellow, means color change is there. So add substrate and measure the color. Color shows the intensity of that particular antigen present in that solution. Then this is the indirect ELISA. Direct sandwich ELISA. Sandwich means what? In the presence of two antibody, there is the antigen is present, or in between two antibody the particular antigen is present, that is called as the sandwich ELISA. That is the antibody is placed over here. First of all, here is the in first indirect ELISA there is the antigen, here is the antibody. Then it is washed solution with the wash with the saline solution. Then antibody, then again there is the addition of the antigen. After that there is the secondary conjugated secondary antibody, which is in a black color, will be added. Then again wash, but it will be not removed. Why? Because in between that two antibody the antigen is present, and that add substrate that measure the color. So add substrate that measure the color, and that color shows the intensity of that antigen. Now third one is the competitive ELISA. Incubate antibody with antigen to be measured. Okay. So first of all, antigen antibody complex is with the mixture with antigen coated well. Then there is the antigen, then antibody is present. Then it is again there is the add enzyme conjugated secondary antibody. Then which antibody is suitable, or which the bond between the antigen antibody complex is strong, which is showing the highest degree of that reaction, that is called as the competitive ELISA. And add substrate and measure the color, and that color shows the intensity of this reaction. Now the label antigen against the different epitope of the antigen is labeled. Then unbound antibody are removed by washing, and then this is followed by addition of color substrate and development of color. Intensity of color is directly proportional to the concentration of antigen in the serum. Now the last reaction, that is a competitive ELISA. Competitive ELISA is what? This is the another variation for measuring the amount of antigen is competitive to ELISA. And in this technique, antibody is first incubated in the solution with the sample containing antigen, and then antigen antibody is then added to the with the help of microtiter well. Then the more antigen present in the sample is less, means what? There is the free antibody will be available to bind to the antigen coated well, and then there is the addition of the enzyme conjugated secondary antibody specific for the isotope of primary antibody, and that can be used to determine the primary antibody bound to the well as well as in a indirect ELISA. ओके नाउ नेक्स्ट मतलब हो क्या रहा है यहाँ पे कि स्पेसिफिक एंटीजन एंटीबॉडी तो प्रेजेंट है, लेकिन एंटीबॉडी को भी स्पेसिफिक फर्स्ट जो एंटीबॉडी जो डाल रहे हैं उसको भी एंजाइम कॉन्जुगेटेड सेकेंडरी एंटीबॉडी डाल रहे हैं, सो दैट सेकेंडरी एंटीबॉडी इट इज ऑल्सो स्पेसिफिक टू द प्राइमरी एंटीबॉडी, और इसके कारण एक प्रॉपरली कॉम्बिनेशन हो रहा है एंटीजन विथ एंटीबॉडी एंड सेकेंडरी एंटीबॉडी, that is indirect ELISA. Now the next process is the immunofluorescence. Immunofluorescence means what? So fluorescence is the property of observing light in a particular wavelength and emitting the light with the different wavelength. मतलब एक frequency जो रहेंगे उससे वो एब्जॉर्ब करेगा लाइट और पर्टिकुलर वेवलेंथ एक पर्टिकुलर वेवलेंथ पे एब्जॉर्ब करेगा, और दूसरे टाइम जब वो इमिट करने वाला है या रिफ्लेक्ट करने वाला है रेस तो उसका वेवलेंथ अलग रहेगा. सो फ्लूरेसेंट डाइज शो अप द ब्राइटली अंडर हाई यूवी लाइट एज दे कन्वर्ट इनटू द विजिबल रेस. ओके, फ्लूरेसेंट डाइज जो रहते हैं सो उनकी एक एबिलिटी रहती है कि यूवी लाइट को कन्वर्ट करते हैं विजिबल लाइट में. सो कून्स एट ऑल शो द लेबल्ड डाइज दैट कैन बी कॉन्जुगेटेड विद एंटीबॉडी, and these labeled antibody can be used to detect the antigen. So dyes are commonly include like fluorescein, phycoerythrin. So fluorescein is what? It is the organic dye and most widely used for the immunofluorescence procedure. It will absorb, ये 490 नैनोमीटर पे लाइट एब्जॉर्ब करेगा ब्लू लाइट, बट जब ये फ्लूरेसेंस करेगा तो ग्रीन येलो कलर का 517 नैनोमीटर पे ये फ्लूरेसेंस करेगा. देन दूसरा जो है दैट इज फाइकोइरिथ्रिन. सो इट इज द इफिशिएंट एब्जॉर्बर ऑफ लाइट दैट इज 13 फोल्ड ग्रेटर देन फ्लूरोसिन, यानी फ्लूरोसिन से भी ये ज़्यादा इफेक्टिव है. एंड इट इज द ब्रिलियंट इमिटर ऑफ द रेड फ्लूरेसेंस. मतलब ये जब फ्लूरेसेंस करेगा तो ये रेड कलर फ्लूरेसेंस करेगा एंड स्टिमुलेटिंग इन अ वाइड यूज इन अ लेबल फॉर द इम्यूनोफ्लूरेसेंस. Now let's see in a diagram. So first of all, this is the cell with the membrane bound antigen, and then there is the primary antibody will be added. This is the basic reaction. Now in a first direct with the direct method with the immunofluorescence. So what? This is antigen with primary labeled antibody. When specific antibody bind to the specific antigen, there is the fluorescence of light, means it is the direct method of the fluorochrome. When antigen is present, primary antibody is done, then in that antigen antibody complex, secondary antibody with the स्पेसिफिक टू द प्राइमरी एंटीबॉडी विल बी एडेड एंड देन बाउंड. मतलब ये जो प्राइमरी एंटीबॉडी रहेंगी इसको ये सेकेंडरी एंटीबॉडी जब बाउंड हो रही है लेकिन इन दोनों की स्पेसिफिसिटी सेम रहेंगी, देन एंड ओनली देन देर इज द फ्लूरोसेंस अकर. मीन्स व्हाट? इट इज द इनडायरेक्ट मेथड विद द फ्लूरोक्रोम लेबल एंटीजन एंटी आइसोटोप एंटीबॉडी. ओके, एंटीबॉडी तो है ऑलरेडी लेकिन उसके भी अगेंस्ट में एंटीबॉडी आ रही है, सो दैट इज एंटीबॉडी आइज. ओके, so then indirect method with the fluorochrome labeled a protein. So there is the antigen antibody, but there is the protein labeled. Okay, specific protein is labeled with that antibody, and then there is the specificity in that antigen antibody will occur, and there is the bond is fixed. So there is the then and only then there is the fluorescence of light. So the third reaction, that is the indirect method with the fluorochrome labeled protein A. So there is the direct and indirect immunofluorescence. Now next there is the application of antigen antibody reaction. So what are the application of antigen antibody reaction? The chief use of antigen antibody reaction are for the blood group detection, and blood group detection for what? For the blood transfusion. Then other serological reaction. Serological reaction we are already discussed. The blood group or whatever the blood reaction occur outside the cell or in vitro condition. Okay, so that is called as the serological reaction, and that are certain to exposure the infection against like Vidal test, ELISA. Okay, then immunoassay for the quantification of various substrate. Quantification means what? There is the detection of what is the quantity of that particular antigen or antibody present in that solution or serum sample. Then to detect the presence or absence of protein in the serum. Then last one is the determine the characteristics of certain immunodeficiency in immunodeficiency disease. Means what? These are the various application of the antigen antibody reaction. Okay. Now in next lecture we are discuss about the MHC molecule, that is major histocompatibility molecule. Now thank you.